introductory slide. This is the famous Drake equation, which everyone in the room knows about. And we're focused on the sort of the FL fraction of suitable planets on which life actually appears. And this is sort of where our work is. Where can life start? So that's another way of saying fraction of worlds in the Drake equation. And what I'll do now is pass it over to Dave, who will take it from here in section 1.
Okay, Bruce, I'll say next whenever I am ready for the next slide, okay?
Gotcha.
So errability is a new term. We made it up because we realized in our conversations that habitability had certain limitations. And we're going to talk about those limitations over the next few minutes. Habitability has to do with which world can sustain life. But errability, we felt, was a word needed to describe worlds on which life can begin. And this gets pretty complicated. Just sustaining life basically means you got some water and you got some sunlight and you got some nutrients and so forth. And then a typical world would be able to sustain microbial life, at least, because microbes simply don't need all that complex, say, an environment to thrive. An arable world, however, is quite a bit more complex. And my time, I'm just going to take you through the complexity as we've brought it together in this paper that we'll be getting to. And that's, I think, is be fun for you to spring this term on other people. You know, say, well, you know, I don't think Venus is arable now. And they'll say, well, it's pretty horrible on Venus, but it might not be arable. So we're thinking that it'd be just great if this word turns out to be a useful word as we have found it as well. Next slide. So this is published with Bruce and myself. Bruce and myself and Francesca Carey, who joined us in the writing effort. And the title of the paper is A Property of Planetary Bodies That Can Support an Origin of Life. It was published in July of this year, and you can see the cover of the astrobiology issue. And the word, by the way, comes from a combining term, you are, which is from the ancient language having to do with original or earliest or primitive. And a very well-known zoologist, Ernst Haeckel, who was working in the mid-1800s, actually used a word like ours, Urschleim. He was German, and that means the primordial slime that he thought was on the bottom of the ocean. And he just proposed that that's where life could begin, in the Urschleim. So keep that in mind as we get a little bit further. There's a real word in the sense that it's made up of two words, one of which simply means primitive or first. So there we are. The authors are myself and Francesca and Bruce, and we just did this mostly by email back and forth. And it's really quite interesting to have this conversation, because as you have a scientific conversation, your brain gets stirred up and ideas come out of nowhere and you can throw them back and forth. And that's why science is such a collaborative effort. I hardly know any scientists these days who work on their own. A typical scientist in my field has anywhere from 4 to 6 grad students and maybe a postdoc, perhaps a technician, and they support their work with grants that they have to compete for. And our little group then just has 3 people kicking this idea around. And what you're going to hear is a result of that conversation. Next slide. So habitability versus errability. Now I figured out a way to really demonstrate that by starting with a flask filled with a solution of organic compounds, including amino acids and sugars and carbohydrates and minerals and vitamins and so forth, everything that a microbial population needs to grow. So there it is, it's stoppered, it's sterilized by autoclaving, and I'm going to put one little bacterial cell into that flask. And if I come back a couple of days later, it is filled with bacteria. So this flask is defined as habitable, but it is not herbal. I could just wait as long as I care to, and we would never see life get started in a flask. So it can be habitable, but not herbal. And we can imagine planets like this. A planet with water and energy and so forth might be habitable, but there might be something missing that is required for life to begin. So that really tells the story, why we felt this new word had to be put into our language of astrobiology. Of course, Louis Pasteur did the same experiment over 150 years ago and showed that, in fact, you really did need dust carrying bacteria to get life started in a nutrient solution like this. Next slide. So let's think now about how we're going to define herbability. We've gone through the definition as a fairly abstract level, but now we're going to get down to the physical and chemical properties required for a planet to be herbal, and also energy sources and combinatorial processes. All of those are part of what we mean by herbability. So it is not just a single condition or even a set of conditions, but it is a set of conditions which in combination create a window of opportunity for life to emerge. For example, next slide. Let's suppose that we have some dry sand. Well, life isn't going to start in dry sand, so let's make it habitable. Let's add some water. And believe me, if there's a few bacterial spores hanging around in that sand, they would start to grow because bacteria don't need much in the way of nutrients to make a living. So a little bit of water added to the dry sand would make it habitable. However, it's still not herbal because we could wait as long as we want and life would never begin there. So let's add now a mixture of organic compounds, just as we added such a mixture to that flask. Well, still, nothing would happen. We could wait around, and as long as a bacterial cell didn't get into it, nothing would start up in the way of life in just with wet sand and water and organic compounds. Okay, let's add an energy source. Now we're getting closer to herbability, but we haven't reached it yet. We could shine a light on the sand, for instance, and the light would, in fact, warm it up a little bit. It could potentially be an energy source, but only if there is a pigment there. They could capture that light energy and turn it into chemical energy. And that's how all metabolism begins on our Earth, of course. You've all learned in basic biology that we live on sunlight. The absence of the sunlight as an energy source, there would be no life on Earth, at least to the extent that we have it today. So we're still not to herbability. So let's add one more process, polymerization. Let's say that some of that energy somehow got polymers to form in that wet sand from those organic compounds. For instance, we might have amino acids there, and they could form simple peptides, or we could have nucleotides there, and they could form short strands of nucleic acids, given the right energy sources. But life still would not start there because there would be no way for evolution and selection to begin. So we're going to add one more item to this. We're going to add encapsulation by membranous compartments. And when we put all of those things into microscopic protocells, we're on the way to urability, because those protocells are all different from one another. They're not all the same, and that means that each of them is a kind of a microscopic experiment, some of which are going to be fragile and just break up, but others are going to be more robust, and they're going to have a particular combination of polymers and energy sources and nutrients so that they can begin to undergo selection and evolution. So that is really what it takes in a very general way to define what we mean by urability. What we're going to do now is to go into further details. And this is where it really got interesting in our conversations. Suppose that we pick out certain parameters and think about the extremes of those parameters and then something in the middle. So look at the part A off to the left, and you can see water either frozen or boiling or someplace in between. So we know that life could not begin in freezing water. Probably life could not begin in boiling water. It's hard for anything to live in boiling water except some thermophiles that learn how to stay alive at 90 to 100 degrees. But you can imagine that life today exists in that urable center. That's where we still thrive, all the way from just a little bit above freezing. There's some algae that survive on snow, in fact, and all the way up to the thermophilic bacteria that survive in hot springs that Bruce and I visit to do some of our experiments. Then we look at light. You can have no light at all, dark, or we can have too much light. There's too much UV light, that's going to break down organic compounds. So again, there's an herbal center where you have just about the right amount of light for life to make a living. Then finally, it can be perfectly dry, like that dry sand, or soaking wet, and someplace in between. And this is where Bruce and I and some of our colleagues are now beginning to explore. There is an in-between of dry and wet, where we are cycling between wet conditions and dry conditions. And it turns out that that in between with cycles of wetting and drying has chemical energy that can drive polymerization. So this is a relatively new way to think about things. That's really the simplest kind of energy you could imagine, just making things wet and then letting them dry out. But thermodynamically and kinetically, we have shown that in fact polymerization can occur as long as you have cycles of wetness and dryness. If you're interested in more details of that, ask me a question afterwards. I can tell you how it works. Then off to the right in part B, we've taken a bunch of ionic solutes. Ferrous chloride is the horizontal line, then going around clockwise, magnesium chloride, sodium chloride, and calcium chloride. And they can either be saturated concentration or they can be fairly dilute concentrations. Taking sodium chloride as an example, it's not quite saturated in the ocean, but it's halfway there. Ocean water is half molar, and saturation is 5 molar sodium chloride. Some halophilic bacteria have learned how to live in saturated conditions of sodium chloride, but I doubt that life could begin in these conditions. There's some very interesting biophysical limitations to trying to live in a saturated sodium chloride. And again, it can't be too dilute either. For instance, iron chloride, FeCl2, is something that Francesca is doing research on in her master's thesis. And she has pointed out in her thesis that Mars is red because of ferric chloride. That's FeCl3. But at the beginning of Mars history, there was no oxygen. and the iron would have been in the Fe+2 or ferrous form. So Francesca tried various concentrations of ferrous chloride trying to simulate the Mars condition 4 billion years ago. And she found some really interesting things about what divalent cations like iron 2 and magnesium and calcium, those are all divalent cations, and they really do have a surprising effect on the ability of membranous compartments to self-assemble. If there's too much of those cations, it's not going to work. Everything just clumps up and falls to the bottom. So getting back to the text here, Goldilocks zones. Habitable worlds are, use this word, that there is a Goldilocks zone in which a planet is the right distance from its star so that water can exist for long periods of time in a liquid state, not too cold and not too hot, just about the porridge, very much like the porridge that Goldilocks happened upon in the bear's home, if you remember that story. So not too hot, not too cold, not too dilute, not too saturated, not too light, not too dark. We're talking about errable centers, and that's that combinatorial aspect of errability that we think is part of our definition. Okay, I'm getting down to my last few slides. Next slide. We're going to talk, I'm not going to go through all of these. I'm just going to pick out a few. And you can see this long list of geophysical factors. There's some obvious rates, just like the size of a planetary body. If it's too small, it cannot maintain an atmosphere because it does not have sufficient gravity. And that makes a real difference. So there's got to be a minimal size at which an atmosphere can remain and take part in the origin of life. Then halfway down, you see number 5, hydrological cycles of evaporation precipitation. That's those wet-dry cycles that we think are so important. Then down at the bottom, you have the presence or absence of subaerial pools or submarine vents. The aerial pools would be on volcanic land masses rising above the ocean. Submarine vents, you've probably heard of, are these hydrothermal vents that are down in the floor of the ocean. So those are 3 of these 12 factors that are geophysical that we have to consider in terms of their effect on errability. Next slide. There's a bunch of geochemical factors. For example, you better not have much oxygen around. Oxygen is very reactive, and it will oxidize all the things that are needed to be present for the origin of life. So that's one of the geochemical factors. The atmosphere must be anoxic. Down to number 8, amphiphilic compounds. Those are soap-like molecules that can self-assemble into vesicular boundary membranes. If you don't have membranes, you're not going to be able to have cellular life. So that is essential for the origin of cellular life. Then down at the bottom, you can see selective processes that lead toward homochirality. Homochirality characterizes all life today. We use L-amino acids and D sugars in our biomolecules. And if you have racemic mixtures in which you have both chiral versions, they're called enantiomers. If you have them both together in a mixture called a racemic mixture, you really can't make functional biomolecules by polymerizing those. It has to be homochiral. But we don't know how homochirality came to be on the early Earth. There's got to be an answer. And then if you get into that, you could spend your life trying to figure that out because it's a very important problem. Next slide. Then these combinatorial factors, this is a slide that Bruce put together. It shows that we can combine polymers with membranes in such a way that the polymers provide stability of the membranes. That's S in that a little diagram under A, or they can penetrate through the membrane and produce pores. They can act as catalysts and enzymatically speed up metabolism. And they can, of course, replicate. If they can replicate, they can sort of pass on genetic information. So those are combinatorial factors that have to be present if we can have an origin of life, an early evolution. Okay, next slide. So we give you now an example of errability in a real planet. Early Mars was errable. There was water. There was very likely to be organic compounds raining down from extraterrestrial dust and meteorites and maybe even some comets. There's light energy from the sun. So early Mars very likely could have given rise to life. And I'm not going to be terribly surprised if we don't find, if we find evidence that there was microbial life on Mars. But Mars today, at least the surface, is inerrable because life could not begin. It's like that dry sand. It would not really be able to give rise to life. So there's a use of the term errable and errability. And we're finding it very easy to work with in our own conversations. And I hope that you have picked up what we're trying to get at with my conversation. Okay, Bruce, I think this is where you might take over. You still there, Bruce?
Yeah, I'm here, yeah. So yeah, finishing up this this comparison, Dave. So Mars and Earth would have been errable 4 billion years ago, but Mars became inerrable. And I think you also pointed out in the paper that today, the question would be today, whether Earth is errable today. And I'll leave that question for later because that's a wonderful discussion. And it also goes back to Charles Darwin. So Mars may still be habitable in rock refuges, but certainly it's possibly not even habitable at the surface. So we're going to now jump into the updates. I promised you some updates on our empirical testing of the hot spring hypothesis for an origin of life. And as Dave referred to earlier, there are two proposed sites. There are several proposed sites for life's beginning, but the two ones that are getting the most work done are hydrothermal vents in mineral membrane cavities, as proposed by Mike Russell just about 30 years ago. And based on the discovery of the black smokers and white smoker hydrothermal vents in the bottom of the ocean, and hydrothermal fields, as opposed to hydrothermal vents. These are hot springs on land with meteoritic organics and protocells. And just before we're not going to focus on hydrothermal vents, but just to point out that hydrothermal vent scenario is really about generating the organics in a sort of continuous stream using gradients that are purportedly available at the vent. So it's one or two steps down from where we're working. We're working at the level of protocells, assembled superstructures of amphiphiles and other or containing organics that form polymers. So really we're in different phases of how life would begin. So Charles Darwin, when asked about the question of life's origins, wrote in 1871 in a letter to his friend T.J. Hooker that he felt that if, and oh, what a big if we could conceive of some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, etc., present, that a protein compound was chemically formed, ready to undergo still more complex changes. And this is an amazing prescient vision by a 19th century great scientist, because what he's describing is that within this environment, a protein compound, a polymer, must be able to form and become more complex and perhaps become longer, develop functions, be selected and amplified. So 19th century science didn't really understand about nucleotides and nucleic acids. That was to come in the 20th century, but this is basically he's describing an away from equilibrium system. And even in his statement, undergo still more complex changes implies a cycling system. So it's pretty breathtaking. So this is an environment which we did a computer animation of to show you the environment we think life could begin: a Hadean volcanic landscape, a hydrothermal spring, a geyser going off, and filling the pool, which is drying down at its edges. So that's just a little peek into that type of a landscape on the early Earth or other Urable worlds. And the sources of organics have always been a bit of a conundrum for origin of life. In terms of hydrothermal vents in the ocean, they're trying to figure out how to fix CO2 and create organics like amino acids from the building blocks. And this has turned out to be very, very difficult. But all the last 30 years, the discoveries in terms of meteorite content, this is a piece of the Murchison meteorite, which Dave worked with in the 80s. And when they ground up a piece of this meteorite, which is a carbonaceous chondrite that fell on Australia in the 1960s, and placed it into solution, membranes formed. Carboxylic acids were present and membranous structures were observed. So that was a clue that membranes can come from space, from this infalling material. In the last 30 years, we've had major space missions. This one, the European mission to the comet, which found that 40% of the organic compounds in the dust around the comet were organic. And more recently, this is a little picture of the sort of cosmochemistry cycle. I know some of the students on this call are studying this. But if you take apart IDPs, interplanetary dust particles, interstellar ices, you find that they're just packed with the ability to generate things like amino acids, nucleobases. And recently the Ryugu from Hayabusa 2, the Ryugu samples have come back. They've actually generated a drop of water. You know, this is big news recently. So in some sense, the organics may be coming from the sky in a form of pre-panspermia. And these are not living organisms raining down, but they're the building blocks that can become living organisms. And if these exogenous feedstocks fall into the oceans, they become dissolved, they disperse, and they're no longer available for chemical reactions. So they have to land on the landscape and then become concentrated in small volumes to become participants in prebiotic chemical circuits. So the view of this that has emerged in the last, say, 2 decades is that you have a lot of organic synthesis in dust on dust and meteoritic infall. You also have synthesis in the atmosphere itself. Work has been done to show that organic compounds are synthesized in volcanic emissions. They're synthesized in atmospheres that are affected by a big impact event. And all these organics can accumulate on the land and then undergo concentration in small pools and wet-dry cycling to generate protocells. And on the right, you can see what lipid looks like under the microscope when you're adding water to it. You know, it has amazing properties to encapsulate and move material around. It almost looks alive, even though it's just lipid. So in an experiment that Dave carried out in the 2000s, and this is showing one of his simulation engines, he introduced a CO2 atmosphere to a chamber, brought it up to 85 Celsius, and then introduced hydration and dehydration by little injectors at pH 3. And he could control how many cycles would happen in little vials. And he could introduce nucleotides and lipid. So he could dry them down and re-wet them multiple times. And what was happening between the layers of lipid as the lipid dries down, as the nucleotides, in this case, we're showing adenosine monophosphate, water would be removed from between the lipid and would pull the reaction forward, and a ester bond would form, and you would form polymers of RNA, an RNA-like polymer, in this case, mono A RNA. And this could be done in multiple cycles. And you can see the gel showing the different lengths of polymer that were present. And through a technology invented by Dave's group in the 80s and 90s, a technology called nanopore sequencing, Dave could actually put these basically different lengths of RNA through a nanopore in a membrane in a device that you can hold in your hand, and it would create blockades as the bases came through this specially designed pore, and it would tell you that you were seeing a polymer and tell you what bases were in the polymer. And recently, groups have been doing this type of wet-dry cycling to generate peptides, and in our group, DNA, oligo-T DNA wet-dry cycling. So these polymers can be quite long. Initially, we were sort of seeing things that were in the 40 to 150 mer range, but they can be very long, and I'll show you an exciting image of that later. So here's the cycle. It's a wet-dry cycle, and you can see on the left that you have, during hydration, you have a budding off of vesicles from a layered state of lipid that dries down in the bottom of the pool, the bottom of the vial. When you add water back in, you get budding, and the budding can encapsulate contents. And so you get protocells in aqueous solution that have to remain stable in order to carry their contents down for another cycle. When you get evaporation, and we can see that, we call that a gel phase, the vesicles come close together and form a kind of a sludge, and then they start to fuse. And they would fuse and drop their polymer cargoes together into the layered state where more synthesis or more polymerization can occur. And then another hydration cycle generates yet more protocells with more polymers. And so this is a multiple selective cycle that can run, basically a combinatorial engine that can run and generate many, many different polymers, each of which, or many of which, can be subject to selection pressures, such that out of a background of random sequences emerge functional polymers. And Dave mentioned some of those earlier, some that would function as pores, or others that would function as catalysts. So it's an engine to generate what Charles Darwin asked for, which was a protein compounds or other polymers ready to undergo more complex changes. So perhaps Darwin's little pond, or warm pond, is updated in the 21st century to a hot little cycling pool. So we'll put it all together, a hot spring origin of life. And this is the central figure we've been used for several years to sort of tell the whole story. So synthesis of a lot of organics in space and in the atmosphere, they fall onto the landscape. They can concentrate in different pools of different chemical properties and sizes and pHs. Products can start to form, say, for instance, membranes, and they can get into a common pool that's undergoing cycling. And if enough of the organic products are there, you have an herbal center in the pool, and the process of forming protocells and testing them individually in their trillions can get underway. And then what happens is you would get a transition to a more robust protocell population. And as these are washed or blown with wind out of a source pool into another pool, they can undergo different cycling, have different resources, and the material is transported across the landscape from pool to pool to pool. This is very important for the combinatorial power of pools in hydrothermal fields, because they're all different. And this possibly gives you the chemical complexity you need to get toward the living system. When you get the living system or living world, you get protocells that are now dividing themselves into daughter cells, and therefore there's a system of hereditable traits. There's a clear system of expression of proteins, expression of function, a kind of quasi genes are there. And then it's this march for complexity and robustness. And if you see in 5 and 6, the protocell mats, because they're early microbial communities, are hitting the estuarine environment into saltwater, which is very, very tough on membranes. So they then have to evolve the active pores to survive in a salty environment. And this is one of the major arguments why life can't start in the oceans or in tidal pools. The salty environment is a later adaptation. And then as you get access to the marine shore, you get colonization of much, much more larger water volumes and shoreline environments. And in the rock record, this is what's borne out. This is geyserite discovered by our colleagues in Australia, 3.5 billion year old evidence of a hot spring. This is lacustrine or lakeshore stromatolite, which is evidence of microbial mats at lakeshore environments about 3 billion years ago. And this is a marine stromatolite from a marine shore environment about 2.5 billion years ago. So recently, we've been going out into the field and testing our hypothesis outside of the laboratory. And this was a series of tests we did at a place called Fly Geyser in Nevada, near the site where Burning Man is held. And in fact, the land itself is owned by the Burning Man organization. So we had to get permits from them. And we went out there with the BBC and they were able to capture our work on camera. And this particular geyser is called Little Pot. And it's about a neutral pH, about 60 Celsius, very high silica content. These geysers grow very quickly. And we're able to cycle our solutions of nucleotides and even some amino acids on slides, on open slides, on a little heat plate you saw there earlier, and do some wet dry cycling. And we saw evidence of, when we look at it under fluorescence, we see that it has encapsulated RNA and DNA. You can see they're glowing on the right. And if we look at some other analysis, we see that we have some oligomers, some things that are formed in longer chains. So that we can form protocells in these hot spring environments. We've done similar work at Yellowstone and Rotorua in Australia. And this is the most recent work at Fly Geyser in Nevada. The converging lines of evidence I mentioned earlier, the continuity on the rock record is very exciting. We met our colleagues, Martin van Kranendonk and his whole team from University of New South Wales in 2015 when they took us out to the Pilbara, and we saw the discovery outcrop for this geyserite here, this sort of striated rock, which was the first evidence of a hot spring on Earth, with evidence of microbialites. And then I was out in a second field season and I basically broke rocks for the first time and found these stromatolites in barite at about 3.5 billion years of age. And earlier we showed you a lakeshore. You can see that the stromatolites, the microbial mat, have been actually pushed up and stacked up in these clasts by water action, fresh water action. And so, and also if you continue in Australia, you can find living stromatolites at Shark Bay, growing today in a highly saline environment in Shark Bay at the marine shore. And recently our colleagues did deep drilling in the Pilbara where they went through the surface down into what was clearly hot spring fascia and detected all of the elements that have been proposed as necessary for an origin of life anywhere on any world. They detected those elements in those drill cores. So it's incredibly compelling parallel line of evidence that we've really been very exciting to be part of. This is a new line of evidence. This is from our colleague Tue Hassenkam at the University of Copenhagen, who uses an atomic force microscope to take samples that we send him and samples that he also wet dry cycles actually in the AFM chamber itself. And you can see what we call rings and strings, polymer thick strings of RNA-like products that are observed visually as these tangled mats in the atomic force microscope. And you can find this in scientific reports published in June of this year. So the scientific and public interest has grown in this approach or this scenario. We were on the cover of the Scientific American in August of 2017 with our colleagues from Australia, and they depicted the model landscape and the wet-dry cycling as this interesting spiral pattern, starting with simple protocells, going through wet, dry, and moist phases, and going out to become living microbial communities. It's an article worth seeing. The Astrobiology Journal published our full hypothesis in 2020, and this is open access, and you can simply find this online. It's something we worked on for several years, and that's our definitive scenario with testable predictions and even pathways to falsification. And we invite you to get in touch, take a look at it, and get in touch. This is how we met Francesca. She found this article and she got in touch, and we've been working together for over a year. Lately, there's been an interesting thing going on. There's been dispute, not surprising in science, but between the hydrothermal vent, ocean vent supporters, and people are more focused on land-based environments for the origin of life. In this article in December 2020 in Nature, talked about the water paradox, that if you're in water all the time, any products that are made are broken down. It's a paradox that life needs to be in water, yet to start life, you can't be in water. So you can't be at the bottom of the ocean, for example. And our colleagues had it out in these articles in the last couple of years. And being a kind of historian, minor historian of science, this has got all the hallmarks of a paradigm shift in science that Thomas Kuhn wrote about in the 60s. So the last little section here, and this is, I think, will lead to a lot of questions, the implications into key questions in astrobiology of where can life start. And I got involved in 2016 with the landing site team for the selection of the Mars 2020 landing site. Francesca works on the actual Mars 2020 Perseverance rover. She's been working on that team because I think University of Hawaii has a big component of people on that. But the Spirit rover, sometime around, I think, 2008, discovered a hot spring outcrop toward the end of its mission. And we know that because one of its wheels was no longer turning, and it acted as a trowel and scooped up soil, and under the soil was basically opaline silica, evidence of hot spring centers, which is an amazing needle in a haystack discovery. And this site is probably around 3.7 billion years old. And one of the things that imaged, now they started looking through the hot spring lens, these rocks here, imaged by Spirit, look like digitate silica nodules that you would see in a hot spring environment like El Tatio in Chile, for example. And in those environments, and we've seen these environments in New Zealand where these basically centers are growing, water is being wicked away, and the structure that is growing is influenced by microbes. Its very structure is influenced by microbes. And so we proposed for Mars 2020 Perseverance to go to this site and sample these rocks. We didn't prevail, and it went to a Jezero crater, as you know. But it's still a very compelling site for detection of life, because life is always preserved on Earth. Anywhere on Earth there's a hot spring setting, it's an extremely good biopreservation environment. So the proposal here is that after all this work and all this field work and laboratory work, we proposed in our hypothesis to start life in any environment requires a dynamic process capable of combinatorial selection on informational and catalytic molecular structures. That you have to get things together into compartments that are polymers, species of polymer, that then can undergo processes of selection, just have to do it. And that aqueous carbon, with all the basic building blocks, in cycling pools can support this, so that early Mars and other similar worlds would be Urable, but ocean worlds, which are, very, very big up there in terms of habitability, may not have the sufficient Urable factors. They may not have exposed land masses and wet dry cycling. They may be all aqueous, so they have the water paradox. They may just simply not have enough Urable factors to start life. They may have been inoculated with microbial life at some point in their histories, but they may be inurable yet and habitable, or they may be sterile. And hopefully missions in the next decades will give us some more clues. So finishing up, the question going back to honor Frank Drake, his question is, how common is life in the universe, and how many worlds, how many civilizations that we could contact would we expect to be able to find? And the origin of cellular life factors into the rise of multicellular, more complex life, and therefore into technological species, clearly. So if we're focusing on this transition from this Drake equation factor to the factor of planets which intelligent life emerges, it really matters how hard it is for simple life to emerge. And our response is that, given that we've actually proposed now these 28 factors and these Urable graphs and Urable centers, at least a starting of a language to describe this that we can apply to all the exoplanets that have been discovered or modeled, that it may be that we can start to make some informed guesses on how hard it is to get life started. And one of the things that I'm proposing to colleagues now and then is not a given that as soon as you have a world with a lot of these properties, you have microbes appear. My intuition tells me that the road to a machine, which is a dividing cell, is fraught with a lot of combinatorial challenges and will require a lot of dumb luck, a lot of catastrophes, and a lot of tries and failures to get to the living microbial world. And that microbes are maybe hard, not easy. And if that's the case, then we would actually expect a much lower inventory of complex life to emerge and a much lower inventory of technological intelligent species to be around. So that's a nice big open question for everyone. So I want to thank Sohan Gita for the opportunity to be part of the conference. The second time we've been in the NorCell meeting, and you can reach us through our Biota Institute, where we are sponsoring and supporting students that are working in astrobiology, especially in the origin of life in surficial environments. And these are some of our collaborators around the world. So thank you for your attention.